You know what's wild? Right now, as you're watching this video, millions of microscopic organisms are being produced in bodies around the world that are so bizarre, so fundamentally different from every other cell type, that if we discovered them on another planet, we'd declare it proof of alien life. I'm talking about sperm cells, and trust me, nothing, absolutely nothing about these microscopic swimmers is normal. Welcome back to MicroZoo, where we dive deep into the microscopic world. Today, we're exploring one of the most misunderstood, most extraordinary, and frankly, most alien-like cells in all of biology. Sperm cells break basically every rule we think we know about how cells work. They're half-dead suicide missiles with a single mission. They navigate using chemistry most scientists still don't fully understand, and they're built with architecture that seems ripped straight from science fiction. By the end of this video, you'll never think about reproduction the same way again. So let's shrink down to the microscopic scale and meet one of nature's most extreme cellular designs. Let's start with the most obvious thing that makes sperm cells abnormal. They're haploid. Now, if you remember your biology class, you know that almost every cell in your body is diploid, meaning it has two complete sets of chromosomes, one from each parent. That's 46 chromosomes in humans, arranged in 23 pairs. But sperm? They only have 23 chromosomes total, just one set. They're walking around, well, swimming around with half a genome. Imagine trying to run a computer with half its operating system deleted. That's essentially what a sperm cell is doing. And here's where it gets weirder. This isn't a bug, it's a feature. Sperm cells are literally designed to be incomplete. They need to merge with an egg cell to create a complete organism. They're like biological puzzle pieces, useless on their own, perfect when combined. But the chromosomal weirdness doesn't stop there. During the formation of sperm, a process called spermatogenesis, the cell undergoes meiosis, a special type of cell division that's already strange enough. But sperm cells take it further. The DNA gets packaged so tightly, using special proteins called protamines instead of the usual histones, that it's compressed to about 1/6 the volume of DNA in normal cells. We're talking about fitting roughly 3 billion base pairs into a space smaller than you can imagine. It's like compressing the entire contents of a library into a single grain of sand, except somehow still keeping it readable. This extreme compression serves a purpose. It protects the DNA during the sperm's dangerous journey and makes the cell's head as streamlined as possible. But it also means the DNA is essentially locked away, inactive, dormant. The sperm cell can't really use its own genetic information. It's carrying precious cargo it can't even access. Now let's talk about the structure of a sperm cell, because this is where things get truly alien. A sperm cell has three main parts: the head, the midpiece, and the tail. Simple enough, right? Wrong. Each of these sections represents engineering solutions so radical that bioengineers are still trying to replicate them. The head of a sperm cell is dominated by the nucleus, which contains that tightly packed DNA we discussed. But covering the top 2/3 of the nucleus is a structure called the acrosome, and this thing is basically a biological warhead. The acrosome is a cap-like organelle filled with enzymes, hydrolytic enzymes that can literally dissolve through the protective layers surrounding an egg cell. We're talking about enzymes like hyaluronidase and acrosin that can break down complex molecular barriers. Think about that for a second. The sperm cell carries its own molecular battering ram, loaded with chemical weapons designed to breach the egg's defenses. It's like every sperm is a microscopic special forces operative, equipped with specialized tools for a single mission. If the head is the warhead, the midpiece is the power plant. This section is absolutely packed, and I mean packed with mitochondria, arranged in a tight helical spiral around the base of the tail. We're talking about 50 to 75 mitochondria coiled in a precise arrangement. Now, here's something most people don't realize. Mitochondria are the powerhouses of the cell, right? They produce ATP, the energy currency of life. A typical human cell might have a few hundred to a few thousand mitochondria, depending on its energy needs. But sperm cells? They cram dozens of mitochondria into a space smaller than a red blood cell, creating an energy density that's off the charts. Why? Because what comes next requires absolutely massive amounts of power. The flagellum, the tail of the sperm cell, is a biomechanical marvel that we're still trying to fully understand. This isn't just a simple whip. This is a complex molecular machine composed of a structure called the axoneme, which consists of 9 pairs of microtubules arranged in a circle around a central pair. That's the famous 9+2 arrangement, and it's found in flagella and cilia throughout nature. But here's what's incredible. These microtubules don't just sit there. They have motor proteins called dyneins attached to them that literally walk along the microtubules, causing them to slide past each other. This sliding motion, when coordinated across the entire structure, creates the characteristic wave-like beating of the tail. The sperm's flagellum can beat at 10 to 45 times per second, propelling the cell forward at speeds up to 0.1 millimeters per second. That might not sound fast, but for a cell that's only about 50 micrometers long, that's roughly equivalent to a human swimming at about 8 kilometers per hour. Here's where sperm cells get really weird. They're basically cellular zombies. Let me explain. Most cells in your body are little self-sufficient cities. They have organelles for manufacturing proteins, systems for waste disposal, mechanisms for repair and maintenance. They can respond to their environment, regulate their internal chemistry, and live for extended periods, days, weeks, months, even years in some cases. Sperm cells? They've stripped away almost everything. They have virtually no endoplasmic reticulum, almost no Golgi apparatus, minimal ribosomes. They can barely make new proteins. They can't repair damage. They can't reproduce. They're single-use cells with an expiration date measured in days at most. It's like they've been streamlined to absolute minimalism. Every single structure that isn't essential for their one mission has been deleted from the design. Their cellular cruise missiles launch, navigate, deliver payload, done. But they still need energy, and lots of it. That's where those tightly packed mitochondria come in. Sperm cells can actually switch between different metabolic pathways depending on their environment. In some conditions, they use glycolysis, breaking down sugars without oxygen. In other conditions, they use oxidative phosphorylation, which is more efficient but requires oxygen. Some species sperm can even use fructose instead of glucose as fuel. This metabolic flexibility is crucial because the environments a sperm cell travels through, from the testes to the epididymis, to the seminal fluid, to the female reproductive tract, have wildly different chemical compositions. What's fascinating is that sperm cells seem to switch metabolic modes as part of their maturation and activation process. It's like they have different gears for different stages of their journey, optimizing their energy use for each phase. Okay, so we have these half cells, stripped down to the bare essentials, swimming through an environment that's thousands of times their own body length. How on earth do they find the egg? For a long time, scientists thought sperm just sort of bumbled around randomly until one got lucky. Turns out, that's completely wrong. Sperm cells are actually capable of sophisticated navigation using a process called chemotaxis. The egg and the structures surrounding it release chemical signals, attractant molecules that create a concentration gradient in the fluid environment. Sperm cells have chemoreceptor proteins on their surface that can detect these molecules. And here's the incredible part. They can detect concentration differences across the length of their own cell body, which is only about 50 micrometers long. We're talking about sensing chemical gradients at a scale that's almost unimaginable. It's like if you could smell something and determine which nostril was getting 0.01% more of that scent, then use that information to navigate across an entire city. The sensitivity required is absolutely mind-blowing, but it gets even more sophisticated. Sperm don't just swim in a straight line toward higher concentrations. They swim in a pattern of turns and straight segments, constantly sampling their environment and adjusting their course. Recent research has shown they might use a strategy called clinotaxis. They swing their head from side to side as they swim, comparing concentrations on either side to determine the direction of the gradient. There's also evidence of thermotaxis, following temperature gradients, and rheotaxis, responding to fluid flow. These cells are using multiple sensory modalities simultaneously to navigate a three-dimensional chemical landscape. And remember, they're doing all of this with virtually no cellular machinery for information processing. The navigation system has to be almost entirely mechanical and chemical, built into the structure of the cell itself. There's also something called hyperactivation, where sperm cells change their swimming pattern from smooth, progressive movement to a more vigorous, whip-like motion. This seems to happen when they get close to the egg and helps them penetrate through the viscous layers protecting it. It's like they have a turbo boost that activates at exactly the right moment. Now we need to talk about one of the strangest aspects of sperm cell biology: capacitation. When sperm cells are first produced, they can't actually fertilize an egg. They're not ready. They need to undergo a series of biochemical and physiological changes called capacitation, and this process only happens after they've been deposited in the female reproductive tract. Think about how weird that is. These cells are manufactured with a specific purpose, but they're not actually capable of fulfilling that purpose until they've been exposed to the right chemical environment. It's like building a key that only takes its final shape after you insert it into the lock. During capacitation, the sperm cell's membrane composition changes. Cholesterol is removed, making the membrane more fluid. Proteins on the surface are modified or redistributed. The cell's metabolism shifts. The flagellar beating pattern changes. Calcium ions flow into the cell through specialized channels, triggering a cascade of signaling events. What's happening at a molecular level is that the sperm is being primed for the acrosome reaction that moment when the acrosome cap releases its enzymes to penetrate the egg's outer layers. If the acrosome reaction happens too early, before the sperm reaches the egg, it's game over. The sperm has blown its one weapon and can no longer breach the egg's defenses. So, capacitation is essentially a timer mechanism that prevents premature activation. The sperm needs to spend a certain amount of time in the female reproductive tract, exposed to specific chemical signals, before it becomes fully armed and dangerous. Scientists still don't completely understand all the mechanisms involved in capacitation. We know some of the players: bicarbonate ions, calcium, specific proteins, but the full picture remains frustratingly complex. Let's talk about just how hostile the journey is for these cells. When sperm cells are first deposited, they're in the hundreds of millions, but the vast majority will never even get close to the egg. They'll get lost in the wrong direction, stuck in the mucus lining the reproductive tract, attacked by the immune system, or simply run out of energy before reaching their destination. Oh yeah, the immune system. Here's something wild. Sperm cells are actually recognized as foreign by the immune system. They have to be, because remember, they're haploid. They're genetically different from every other cell in the body. In the testes, they're protected by something called the blood-testis barrier, which keeps immune cells away. But once they leave that protected environment, they're under attack. The female reproductive tract has immune defenses, white blood cells, that treat sperm like invaders. And rightfully so. From an immunological perspective, they are invaders. So the seminal fluid that carries sperm contains immunosuppressive factors that temporarily dampen the immune response, giving the sperm a fighting chance. Even with this protection, the attrition rate is staggering. Of the 200 to 300 million sperm that might be deposited, maybe only a few hundred will make it to the vicinity of the egg. And of those, only one will actually achieve fertilization. The journey can take anywhere from 30 minutes to several days, depending on timing and conditions. During that time, sperm cells are navigating through environments that vary wildly in pH, viscosity, chemical composition, and temperature. They're swimming distances that are millions of times their own body length. If you scaled that up to human size, it would be like swimming from California to Japan in a few hours, while being attacked by the immune system with half your genetic information missing. When a sperm finally reaches the egg, what happens next is like a precisely choreographed molecular dance. First, the sperm must bind to the zona pellucida, the protective glycoprotein layer surrounding the egg. This binding is highly specific. The proteins on the sperm's surface must match up with receptors on the zona pellucida, like a key fitting into a lock. This species specificity is what prevents, say, a human sperm from fertilizing a mouse egg. Once bound, the acrosome reaction is triggered. The membranes of the acrosome fuse with the sperm cell's membrane, releasing all those enzymes we talked about earlier. These enzymes digest a path through the zona pellucida, and the sperm literally burrows through, still beating its tail frantically. Once through the zona pellucida, the sperm reaches the egg's cell membrane itself. Another set of membrane proteins mediate the fusion of the sperm and egg membranes. And then, in one of the most remarkable moments in all of biology, the sperm's nucleus is drawn into the egg's cytoplasm. Almost immediately. We're talking seconds. The egg undergoes changes that prevent other sperm from entering. This is crucial, because if multiple sperm fertilize a single egg, also known as polyspermy, the resulting embryo will have too many chromosomes and won't develop properly. Inside the egg, the sperm's tightly packed DNA begins to decondense. Those protamines that were compressing it are replaced with histones. The paternal chromosomes prepare to meet and combine with the maternal chromosomes, creating a complete diploid genome. A new organism begins to form. And the sperm cell itself? It's done. It's sacrificed everything, its membrane, its structure, its individual identity to deliver its genetic payload. It literally ceases to exist as a separate entity. So there you have it, sperm cells, half dead, half genome, hyper-specialized cellular suicide machines, equipped with molecular warheads, sophisticated navigation systems, metabolic flexibility, and the ability to literally dissolve their way through biological barriers. They're unlike any other cell type in the body. They break almost every rule of cell biology, and they're absolutely essential for sexual reproduction across countless species. The more we study these microscopic swimmers, the more we realize how sophisticated they are. They represent evolution's solution to one of biology's fundamental challenges: how to combine genetic information from two parents to create offspring with new combinations of traits. Nothing about sperm cells is normal, and that's exactly what makes them so extraordinary. If you learned something new today, hit that like button and subscribe to MicroZoo for more deep dives into the microscopic world. Drop a comment below what other microorganisms or cells should we explore next. Until next time, from all of us at MicroZoo, keep exploring the invisible world around you.